Good morning. So first of all, we're really excited to have you all with us today and join us for our webinar. We'd like to welcome you. We'll introduce ourselves shortly. One thing I did want to point out was the number of people that were registered as of about 8 o'clock this morning. This is the breakdown. So we have a huge diversity of different audiences and participants. We can see we have a large number of extension educators or related in academia, as well as lots of producers. So we're so excited to see the split of many producers on here, as well as all these other groups that we have: cottage industries, market managers at 30, processors, restaurants, retailers, wholesale distributors, and then there were a few that were unspecified. So we're really excited to have you all here today and hope that this will be an informative time for you. So who are we? We are a team from Virginia Tech and Virginia Cooperative Extension. We're really excited to share with you. We have Dr. Melissa Chase, who works with consumer food safety, and Dr. Joel Eifert, who's an extension specialist and leads our food innovations program. Dr. Laura Strawn, who's a produce food safety specialist and an associate professor and housed at our Eastern Shore AREC. Dr. Rob Williams, who's a professor and food microbiology extension specialist. And then myself, I'm an extension specialist and also head up the team for the fresh produce food safety in Virginia Cooperative Extension. Today, our speakers will be myself, Joelle, and Robert, and we will be sharing some different aspects. I'll get into the agenda in a moment. And then Melissa and Laura will be actually fielding the questions and helping with the question and answer time. So just as far as an agenda for today, I'm going to provide a brief overview on food safety, kind of set the context. We entitled this Expanding Food Safety Protocol, so building upon all the things that you are doing and how coronavirus impacts that. Rob will be spending some time talking about coronavirus and what that looks like in a generalized way. And then we'll actually, to the heart of this is really how can we expand practices that are related to how coronavirus is impacting our food safety. And we're going to look at several areas that are critical. And then we're going to kind of spend some time on dispelling some of the myths. There's so much information out there right now, and a lot of it is misinformation. And so we wanted to discuss some of those myths with you. And then we'll lead into our question and answer time and then a very brief wrap up. So whenever we think of food safety, some of the things that we always look at first are what are the hazards that are a part of the system that we are working with. We also look at the ways that food can become contaminated. And so what are the means for that? So whether it's water quality, whether it's workers, whether it's the equipment and tools, whether it's the kind of things that we actually put onto our soil and stuff. The hazards typically are grouped as biological, chemical, and physical hazards. Those hazards can be present on the farm as well as post-farm, and we're going to look at that a little bit here briefly. So one of the ways that we've often found really nice to discuss food safety risk is looking at this whole idea of a farm to fork continuum. So when we think about this farm to fork continuum, if we look at every stage of this, whether it's the production on the farm or whether it's aquaculture or greenhouses or animal production, we can see that this flow of food all the way from the farm through the different marketplaces or the processors, the distribution centers, where that food tends to wind up, whether it's institutions such as colleges and universities or hospitals, to retailers, restaurants, or to the home consumer at that end of it, the fork end, that every single part of those, that continuum represents potential for food contamination. So we often, when we look at that continuum, the practices that we put into place are aimed at mitigating those risks along that continuum. So whether it's good agricultural practices or gaps that are on the farm, all of these can pertain to those gaps. And then past the farm, where we're looking at post-harvest handling in these different entities that we just looked at, a key one being worker trainings related to hygiene and health, water quality, animal and pest control, equipment, facilities, and tools, the storage of the product, whatever it might be, and the transportation, and then, of course, marketplace handling, whatever that market might be, or market sector. Now, I know that with all the work that we are doing here in Virginia, many of people that we work with are incorporating best practices, whether they are in the form of GAPs or HACCP plans or any of those best practices that are done. We know that people are incorporating that. So what we really are interested in today is really looking at how the coronavirus is really impacting that. So again, we've got this new beast that we're dealing with. Many of us, all of us have been impacted by it. And what we're going to do now is look at how does coronavirus really play into what we're already doing and trying to get back to some normalcy.
Okay, thank you, Amber. So I'm going to spend a few moments here talking about the coronavirus itself and some of the principles that relate to especially food processing and food manufacturing, as well as food production. But I want to start with a few statements here related to the impact of this coronavirus, the SARS-CoV-2 virus, on the food system. And so these are statements that were made by Frank Yanis, who is certainly an important person at the FDA, the Deputy Commissioner of Food Policy and Response. And he made these comments on April 2nd. He said the U.S. food supply remains safe for people and animals. Okay, so that's number one. There's no thought at this point that coronavirus is making the food supply unsafe. Secondly, there are no widespread or no nationwide shortages of food. Okay, there might be local shortages in certain areas due to supply chain issues, but there are really no nationwide shortages of food, and there's not expected to be any. And third, there is no evidence, this is important, of human or animal food or packaging being associated with the transmission of coronavirus. So just to start out by saying, it doesn't appear as though foods are a primary source of transmission of this SARS-CoV-2 virus that causes the COVID-19 illness. Next slide, please, Amber. So what are coronaviruses? Well, coronaviruses have certainly been in the news a lot lately, but they're not new. We first identified human coronaviruses back in the mid-1960s, and it's known that there are several types of coronaviruses that are common in humans that cause upper respiratory illnesses fairly commonly. There are also a number of animal coronaviruses, but where we see the real issue is where we have new coronaviruses that jump from an animal species to a human. And so that is what has led to the emergence of SARS that occurred back in the early 2000s, the virus that causes severe acute respiratory syndrome, and then also the SARS-2 that we're dealing with now, and also MERS, which is Middle Eastern respiratory syndrome. And so in those cases, animal coronaviruses were transmitted to humans. And generally speaking, the animals that transmitted those to humans were intermediate hosts. They weren't the primary host. We'll look at that in just a moment. So in terms of nomenclature, COVID-19 actually refers to the illness itself. It really stands for the novel 2019 coronavirus disease. The virus itself is referred to as SARS-CoV-2, or severe acute respiratory syndrome coronavirus 2. And so that is the virus that causes the COVID-19 illness. Next. So if we understand how the virus is transmitted, then we can put in interventions to prevent further illnesses. And so this is going to be important just in our general public life, and it's also going to be very important for our employees and ourselves and our food production and processing situations. So understanding this is key importance. The virus is transmitted primarily, it's believed, by the Centers for Disease Control through aerosol droplets. Okay, so this is can be really two major ways. One is a sick person is coughing or sneezing or just breathing, and virus particles come out into the air. They're very small viruses, and they end up getting transmitted in a large airspace, and then people can breathe those in. It becomes taken into the mouth or nose, and even sometimes attached to the eyes. And then that virus can then go in and infect its sort of target cells, which are the cells that present angiotensin converting enzyme 2 or ACE2 sites. And so primarily the cells that are being affected are lung alveolar epithelial cells. So it's not clear at this point if there are any cells in the gastrointestinal tract that are being much impacted by this virus, but certainly those in the lung are being very much impacted by this virus. So the issue is preventing that virus from getting into the upper respiratory tract and the lungs. The second are infected asymptomatic persons. It appears that one of the major routes of contamination now, or major route of transmitting the virus, are asymptomatic persons who are breathing out the virus and therefore or touching surfaces, and that they have surfaces that they've touched that can then transmit the virus to someone else who touches that surface, okay? And so that seems to be a very important way. So just seeing someone who's sick is not enough to know if they potentially carry the virus. So that's an important fact as well. In terms of the sort of second way that the virus is transmitted, it's by surfaces. So aerosols that are coughed or breathed out can settle on surfaces, or someone who is sick touching their mouth or coughing into their hand, or touching their eyes or nose can place that virus onto a surface. Now, viruses do not grow outside their host. So a virus is not going to grow or multiply on a surface. It's not going to grow or multiply in a food. So it sits there and decays and becomes unviable over a period of time. And we'll look at sort of some of the surfaces and what periods of time we're sort of estimating might be the time of viability for this virus. So then for someone else to get that virus, what they have to do is actually touch that surface and then touch their face. The virus has to have an entry point, usually the mouth or nose are the primary ways, and maybe even the eyes. And so from that touching standpoint, touch a surface, viable virus in them. That's considered to be a secondary method by which this virus is being transmitted. Again, the primary is person to person. So the symptoms are fever, cough, and difficulty breathing. Those are the primary symptoms. But some people also have tiredness, aches, and runny nose and sore throat. And so, unfortunately, there are many infected people who are asymptomatic. So again, just avoiding those with a cough or those who have clearly a runny nose or difficulty breathing is not going to be enough to ensure that we are not transmitting this virus. Next slide, please. So one of the big questions is, where did this virus come from? So I already said that we have a number of human coronaviruses, and throughout the population, we have some immunity to those. But when we have these new viruses that jump from an animal species to us, and in this case, this particular coronavirus, then we don't have any immunity. And so it spreads very widely and very rapidly, as we've seen all across the globe. So virologists are pretty sure that this SARS-CoV-2 virus that causes COVID-19 illness is a bat coronavirus. So it's a coronavirus called a beta type coronavirus, but they believe it originated in bats. So the big question is, what animal served as the intermediate host that then allowed that virus to be transmitted to humans? Right now, that answer is unclear. So for the SARS-CoV-1 virus and in these severe acute respiratory syndrome that occurred in the early 2000s, it was believed that bats transmitted the virus to civet cats, and then the civet cats transmitted it to humans, okay? In terms of the Middle Eastern respiratory syndrome coronavirus, it's believed that bats transmitted that virus to dromedary camels that then transferred that to humans. So with the SARS-CoV-2 virus, the one that we're currently dealing with, we're not exactly sure what the intermediate host is. Some of the suggestions have been pangolins, which are these scaled anteaters that are found throughout Asia and also in Africa. And but that was one of the original intermediate hosts that was identified. However, it has certainly, as time has gone on, there's been some question about whether or not the pangolin is actually the intermediate host. Other potential intermediate hosts were snakes and turtles. But in any regard, it's believed that the first crossover may have been with one of these animals in a wet market in Wuhan, China. Next slide, please. Okay, so where do we find this coronavirus in infected individuals? What fluids? So it's known that upper and lower respiratory tract specimens are positive in infected individuals. What may be a little bit less known is that the virus can also be found in stools and in blood and possibly even urine, even though it's believed to be rare. So there are a number of body fluids that potentially can transmit this virus. So it's not just about upper respiratory hygiene, it's also just about overall personal hygiene that's going to be important to prevent the transmission of this virus. So if we know that the primary way that the virus is transmitted is person to person through aerosol droplets, and the secondary way that the virus is transmitted is through contaminated surfaces that someone touches and then touches their face, we can come up with some controls that can greatly reduce the amount of virus that is capable of transferring to someone else. So primary method is going to be social distancing, and you're going to hear all of these controls again because they're so important and things that I know that you've heard. Right now, the recommendation is to maintain at least a 6-foot social distance, and don't spend a huge amount of time with an individual who may have been exposed or may be sick from the illness. Second is to wear a face covering. Face coverings were something that's a recent recommendation from CDC, and we're not talking about masks that are used for healthcare like N95 masks. We're talking about face coverings that you can purchase, such as surgical masks or those that you make at home. And there are certain recommendations that the CDC is now making about how to make those face coverings. But it's been recommended that these face coverings be worn in situations where social distancing is hard to maintain, like a grocery store, okay? Another issue here then, if we move over to the surfaces that we have to be concerned about, or the environmental contamination sources, then a major aspect is going to be hand hygiene. So even if we touch a surface that's contaminated, the only way it's going to infect us is if we move that to our mouth or nose or possibly eyes. So we want to stop that process. We want to intervene there and make sure we have very good hand hygiene all the time, okay? Proper hand washing. We're going to wash those hands for 20 seconds, okay, and do a very good job. And remember, the technique is the most important part. And also the use of alcohol-based hand sanitizers. This particular virus has a lipid bilayer surrounding the genome of the virus, and it also makes it susceptible then to alcohol-based sanitizers. So when we use those sanitizers, we're able to destroy the virus on our hands, so it doesn't become an issue for us. However, the best scenario is proper hand washing followed by the use of a good alcohol-based hand sanitizer. Hand sanitizers are generally not that effective if the hands are dirty or not cleaned well. Okay. The next thing is keep the hands away from the face. This is actually very difficult. So this is something that takes intentional, conscious practice to not do it. Microbiologists just learned this as part of their training. So we may be a little bit better than the general public about not touching our faces. However, as far as individuals, studies have shown that people touch their face on average over 20 times an hour. Okay, so this is something that's going to take some intentional, conscious focus to us stop touching our faces with our unclean hands. And then the next is cleaning and sanitizing surfaces. And so as we go through, Joelle especially is going to be giving you some tips on ways to control this virus, especially in food processing and on some of those environmental samples. Okay, next slide, please. So if we're going to focus then in the food side of control in ensuring that this virus is not transmitted, then a primary area that we're going to focus on is, one, our workers and people we have in our food facilities, and making sure we don't transfer that virus through improper social distancing and these type things, okay? But the second one that's going to be very important for us is making sure that we control the virus on these surfaces, okay? Various surfaces that could be contaminated by aerosols or a sick person touching the surface. So there have been some studies that have been done over the years on various coronaviruses that can allow us to see a little bit about how this virus can persist on surfaces. The most recent was published on March 17th of this year in the New England Journal of Medicine from scientists from the NIH, and it actually studied the SARS-CoV-2 virus, the virus that causes COVID-19, the illness that's plaguing us right now. They found that the virus can remain viable on various surfaces like plastic, stainless steel, and cardboard and copper for varying lengths of time. They found that on plastic that the virus can potentially remain viable for up to 72 hours. For stainless steel, it looks like right about 48 hours is the maximum length of time that this particular virus stayed viable, but it could be as up to 72 hours. Okay, so it's really sort of there in the middle. Cardboard is 24 hours, and copper was somewhere between 4 and 8 hours. Now, in terms of aerosols, it appears that the aerosols themselves can linger in the air for potentially up to 3+ hours, but over time, these aerosols do settle. So in the breathing space, depending on how tall you are, in the breathing space, the time where that might be an impact is determined sort of over a period of time. Okay, next slide. Okay, so I gave you the study on persistence on surfaces that was recently published on the SARS-CoV-2 virus, the one that's causing COVID-19. Now let's take a look back at some comparative studies that were published this year in terms of the other coronaviruses, like the MERS coronavirus and the original SARS coronavirus, and then just other human coronaviruses, to give you a sense of sort of what historically these have been shown to survive, how long they've been shown to remain viable on surfaces. Now, I want to be clear about this. These studies primarily looked at viruses that were viable. So they looked at cell culture as the method for determining viability, okay? So you may have heard that there's some question about, well, detecting the RNA of the virus doesn't really tell you if it's viable. That's true. Okay, so studies that focus on just detecting the RNA of the virus on the surface don't necessarily tell us if the virus is viable at all. It could be non-viable at that point, non-able to cause infection. But for those that are still on the surfaces, what we want to be able to see is the ones that have been able to show infectivity through cell culture, and that's sort of what this data is looking at. So this particular study was a review of several articles from various from various studies. And so just to give you kind of a sense, a couple of things I want you to look at here. One is the type of surface and the impact of that type of surface. And the other one is looking at the temperature. Okay, so most of these studies were performed at room temperature. You'll see that in a minute. But for steel, for example, the MERS coronavirus was shown to be able to persist for up to 48 hours. Okay, so that's about what we see with the SARS-CoV-2. For aluminum, 2 to 8 hours. Okay, wood up to 4 days, depending on the type of wood. Paper, and basically what we're talking about, where this virus, how long does it persist on these? It really depends on several factors, like how dry the paper wood is and various factors. It does seem that humidity increases the length of survivability of the virus itself, and dryness in some cases reduces the viability, but several factors have to come in play. In terms of glass, we're looking at potentially 4 days on that surface. Okay, next slide, please. Plastic. Certain studies have shown that plastics are less than or equal to 5 days for SARS viruses. For the MERS virus, it was about 48 hours. Okay. In other studies with certain strains, you've seen up to 4 days of plastics with this original SARS virus. And in terms of other aspects like fabrics, like surgical or rather disposable gowns and these types of things, potentially up to 2 days. But again, it depends on, it's very difficult to determine on various surfaces, whether it be fruit and vegetable surfaces or clothing surfaces, just how long this virus will survive. It appears not a tremendously long time. So it's also an issue of the contact of that virus with that surface. How easily is it going to be released? Is it really an issue? Even if it is viable, is it going to be released from that surface? And from that surface, is it going to be able to make someone sick by being transmitted from that, or is it going to be firmly affixed to that surface? And so note that many of these cited studies, in this particular study, were performed at room temperature. Okay, so it's expected that higher temperatures are expected to result in quicker inactivation of the virus, whereas cooler temperatures are expected to extend persistence. So a refrigerated item that had been touched by a contaminated person might be able to extend the survival of that virus for a longer period of time. It's also believed that freezing may preserve the infective, viable virus pretty much indefinitely. Okay, so those are differences in temperature, right, that as an impact on the virus survivability on surfaces. Okay, so let me take a look at a couple questions here. Okay, so one of the questions is, we talked a bit about humidity. It does appear that humidity improves survival, the viability of the virus, but exactly how much, under what circumstances, and how that relates to the surface they're on, and those factors is not very clear. Okay, and then in terms of how long does the virus persist on fruits and vegetables, we don't know that right now. We do not think that fruits and vegetables are an important source of the virus as far as a way of transmitting the virus to a person. However, we also recommend that people wash fruits and vegetables well, just with running water, and that seems to be a intervention that's deemed to be acceptable by the public health agencies. Okay, so I think I addressed the other. Okay, next slide, please. Okay, so the next phase in, if we know that the virus can persist on surfaces at varying lengths, then, and we want to break that chain between environmental contamination and then someone contaminating themselves with it, then we need to talk about biocidal agents that are going to be able to inactivate this virus. Okay, so ethanol, most of the hand sanitizers that are being used are alcohol-based, and many times it's ethanol. And so studies have shown that ethanol is pretty effective at destroying this virus. Within 30 seconds of exposure time, when you get to 78% based upon these studies, or at least 10 minutes of exposure time for 70% ethanol, you get a significant reduction. So that's why ethanol has been recommended as a good hand sanitizer, as long as the percentage, and it's been quoted as different as 60+% or 70+%. In terms of hydrogen peroxide, it appears that hydrogen peroxide is very effective at least 0.5%. Okay, sodium hypochlorite, so bleach has variable effectiveness depending on its concentration, and again, how it's applied. Remember, bleach is rather easily quenched by organic materials, so using bleach in a good way, an effective way, takes a bit of knowledge and planning. But it does seem to be effective at concentrations that are higher than what we would typically use in food processing facilities on food contact surfaces. Next slide, please. And then in terms of some quaternary ammonium compounds, there seems to be variable response to quaternary ammonium compounds. So with benzalkonium chloride, we see that there have been studies that show that 0.2% for human coronaviruses was not very effective at all. Okay, so for mouse hepatitis virus and for canine coronavirus, it seemed to be much more effective, but there really just hasn't been quite enough work on certain quats to know just how effective they are. Interestingly enough, you're going to see a number of these quats that are represented on the EPA's list, the N list of virucidal agents. Okay, so be sure to look at that document. You'll see that later. I want to make a statement here that vinegar has not been shown to reduce viral infectivity to an acceptable level. Okay, so there have been a number of folks who've suggested using vinegar as a sanitizer. We have no evidence that vinegar is able to destroy this virus or render it non-viable. Okay, so also we have a number of products that are peroxyacetic or peracetic acid products, and to my knowledge, there's just not much data that's available on whether or not these peroxyacetic acids are products are effective for destroying the coronavirus. Some suggestion would say yes, but the issue is we just don't have the science to back it up at this point. Okay, next slide. So another aspect of control besides the biocidal agents would be, okay, what about heat? At what temperature can we destroy this virus? And so the French Agency for Food, Environmental, and Occupational Health and Safety has stated that, as with other known coronaviruses, this virus is sensitive to cooking temperatures. Heat treatment at 63 degrees C, or approximately 145.4 degrees Fahrenheit for 4 minutes, can therefore reduce contamination of a food product by a factor of 10,000. And so that's considered to be acceptable. So the French Agency for Food, Environmental, Occupational Health and Safety says 63 degrees C or 145.4 degrees Fahrenheit for 4 minutes. Next slide, please. Another study that was performed or reported back in 2004, which certainly doesn't include the SARS-CoV-2 virus that we're talking about, but similar viruses, the original SARS-CoV-1 virus now, showed very similar results. We saw that at 56 degrees C or about 132 or 133 degrees Fahrenheit, we see that the virus viability was reduced to acceptable levels before 20 minutes. With 65 degrees C, we saw that viability was reduced about that same range, somewhere around 4 minutes is where we see that the viruses were rendered inactive at 65 degrees C again, or 149 degrees Fahrenheit. So at 75 degrees C or 167 degrees Fahrenheit, the way the study was done, it ended up showing that within 15 minutes, because this is sort of the way that the study was done, within 15 minutes, certainly the virus was rendered non-viable. So you expect higher temperatures are going to result in quicker kill of the virus. Okay, let's take a pause here for just a moment for any questions, and I will try to, let me see. All right, so I'm checking here to see what questions are coming in.
Rob, if you have your phone, I've sent you all the questions that are specific to your section.
Gotcha. Okay, so we're going to talk about chlorine a bit later. Actually, Joelle Eifert's going to talk about chlorine, so I want to address that here. She will talk about levels that have been recommended and how those relate to the food processing environment, okay, or food handling environment. And so the question about oxidate, again, and peroxyacetic acid, we don't have the data at this point that I'm aware of where studies on these particular coronaviruses have been performed. So based on the fact that peroxides are quite effective, and you may suggest that if a study was performed, it may show that oxidate was very effective. However, we just don't have the data to say that, okay? And so with alcohols, whether it's isopropyl or ethanol based, it's recommended, again, to get in that 60+% range, and some say even 70% of the alcohol in the solution. Okay, so at this point, I'm going to turn it over to Joelle Eifert.